Hello and welcome back to Enveloped Negative-Strand RNA virus part 2. Today what we are going to talk about will be the life cycle of Newcastle disease virus. So this is the cartoon that shows the life cycle of Newcastle disease virus, whereby the virus initiates the infections by attachments, then fusions, and from here we know that the RNA virus has a negative strain genome that it needs to produce mRNA for the viral protein productions, and then after that we'll look at how the virus replicate their genome, and then assembly, and eventually egress from the cell. So we are going to look in detail each of these steps in the life cycle of Newcastle disease virus. So to initiate the infections, first is the virus need to attach to a receptors on the host cell. And what we can see here is this is the receptors for NEV, which it contains something called salicylic acid. So heme agglutinin neuraminidase protein is the one that responsible for binding the virus to these salicylic acid receptors. And we know that there are many cells in our body or in the animal's body has this salicylic acid on the cell receptors. Alright, so first HN will bind to the salicylic acid receptors on the host cell. Now there are two schools of thought on how these interactions play, whereby the virus, how the virus enter the cells. So the first model say the HN and the F protein, which is on the surface of the virus, HN and F, in first model they are not interacting with each other. Whereas in the second model, they say the HN and the F is always interacting with each other. So both of these ultimately wanted to make the virus to enter the cell. Regardless of this or this, the outcome will be final fusion protein will fuse the cell. But let's look at how these two models come up with. So the first model say the HN and the F originally they are not interacting. So they are on the surface of the virus, but they are not interacting with each other. That's why they are separate. Alright? But when HN protein binds to the receptor, salicylic acid receptors on the host cell surface, these interactions causes a conformational changes on the HN protein. So you can see original HN protein, but when they interact with the salicylic acid receptors, it causes the conformational changes on the HN, and which then expose this portion of the HN to allow the F protein to interact. So this is what we see here. After HN protein binds to, then it have a conformational change and expose a certain region of the HN that allow F to bind to it. Alright? So what about the second model? The second model say the HN and the F are originally in contact with each other. But when HN interact or bind to the receptors, salicylic acid receptors, this causes that the HN and the F are being separated. Alright? So they are separated. So first they are combined, they are together, but once the HN binds to the salicylic acid receptor, conformational changes, which causes the F to be separated from the HN. Alright? So remember, when protein and protein interact with each other, they will cause conformational changes. Here you have HN and F. After HN binds to salicylic acid receptors, conformational changes that allow F to bind to it. Now, when F binds to the HN, that will cause conformational changes to the F as well. So what happened here is that F now has conformational changes, and same goes for this one. Originally, they are interact with each other when HN binds to salicylic acid, and this causes the F to be separated, and because the protein originally are in certain conformations, when they are separated, the F will also undergo conformational changes. So regardless of this model or this model, ultimately what happened to the F protein is the F protein will now have conformational changes. Originally, their fusion peptide the F protein has a fusion peptide. And if you remember from dengue virus, you know that fusion peptide is very important for the fusions of the virus. So now F protein has the fusion peptide that being exposed. And what happened now is the fusion peptide exposed and it poke or penetrate the cellular membrane, which is green color. This is viral membrane. So showing here is the F protein. The red color here is the fusion peptide. So now fusion peptide is hydrophobic and it can penetrate the cell membrane. And remember I mentioned earlier that F protein, when their fusion peptide is being extended, it behaves like a spring. So here is all the alpha helix bundle. Okay, alpha helix bundle, they bundle together. So this make the F protein become springy, extend, and now the F protein string want to come back. And what happened here? It pull the membrane from the cell, getting closer and closer, and eventually the lipid fuse together, and this allowed the nucleocapsid of the NDV to be released into the cell. So this is how the virus first undergo attachment, then fusions. So you will notice that when you study envelope virus, if you understand dengue virus, the same concept, you can use it to apply for other envelope virus. For example, in this case, Newcastle disease virus, attachment and fusion. So what happened next is after the fusions of the two membrane, it will now release the NP, the P and the L and the RNA into the cytoplasm. Now this diagram is a TEM diagram of the NP protein. What we can see here is the alpha helix, the nucleocapsid that is in the helical shape. Alright, this is all the NP protein in the helical shape. Alright, so going in, this is the portions that go into the cytoplasm. Now for NDV, the RNA, the NP protein, the P protein and the L protein will remain in the cytoplasm. For dengue virus, the RNA is being released from the capsid and moved to the RAF ER. For NDV, it remains in the cytoplasm. Okay. Now this complex is now called the transcriptase complex. Alright. From the name, we can see that the enzyme, it is an enzyme that carry out the transcription, transcriptase complex. Alright. So why is it so important for the virus to carry the enzyme? Alright. Unlike poliovirus, unlike dengue virus, they only release their RNA. They don't even need to carry their own RNAs. Okay, the the polymerase. The polymerase is encoded in their genome, but they do not carry the protein. Whereas for NDV, negative-strand RNA virus, they need to carry their own polymerase protein inside the virus. That's why the polymerase for NDV is structural. Okay. These are all the structural protein. Alright? So once it goes in to the cytoplasm, like I said, third step for virus infection, attachment, fusion, the third step will be synthesis of viral protein. So in order for the virus to synthesize their viral protein, what it requires now is to produce mRNA. Alright? So this will be the classical translation process where the RNA for NDV, it has a cap and it has poly-A tail. Alright, it's the classical the translations of the mRNA. Alright, so what we can see here is this is the genome of NDV, and at the 3' end, this is the negative strand, so you have 3' end here and 5' end at the other side. Okay, unlike positive, you have 5' end and 3' end. This is negative strand RNA virus, so the genome is 3' to 5'. And what we can see here is at the 3' end, there's a region called the leader sequence. Alright, the leader sequence. Now this is the regions that does not code for any gene, right? It's simply as a sequence of RNA that allow polymerase to bind to. Alright, so it also behaves like a promoter for the virus. This is where the polymerase bind to the leader sequence at the 3' end. We also have another end of the genome, which is the 5' end. We call this the trailer sequence. Alright, this is where the RNA polymerase fall off. Alright, so encoded in the genome, we have 1, 2, 3, 4, 5, 6. Represent this 6 structural protein of the virus. So remember I mentioned to you that the important gene or important protein will be at the you will first encounter the important gene, and then the least important will be at the end. Right? So the NP protein will be the first gene to be translated or transcribed because it is very important to protect the RNA. So we have NP gene, P gene, M gene, F, HN, and L. Glycoprotein, nucleocapsid protein, and the polymerase protein. Right. So what we can see here is in poliovirus and dengue virus, I always use this to refresh your memory. In poliovirus and dengue virus, you only have one ORF. Means that what one ORF means is you only have one start codon and one stop codon. It will synthesize the gene in one long polypeptide, and it being cleaved to individual protein, so that it becomes one protein, one protein, one protein, one protein. You only have one ORF. Whereas for NDV, even though the RNA is non-segmented, it encodes 6 genes, where here we have 6 ORF. Alright, 6 ORF means that MP has its own start codon and stop codon, P has its own start codon, stop codon, M start, stop, and so on and so forth. So meaning that individually, this would be one mRNA, one mRNA, one mRNA, one mRNA, one mRNA, and one mRNA. So each of these genes is one mRNA with a cap and polyacyl, a cap and polyacyl, a cap and polyacyl. Alright? So, showing here the genome. On the top of the genome, we can see there is a number 2, 1, 1, 3, 47. So what this number represents is the space or the nucleotide in between MP and P. So what it means here is for the gene of MP and the P to be separated, there is 2 nucleotides which doesn't belong to MP or doesn't belong to P. So this one we call the intergenic region, okay, or intergenic sequence. For P and M, there's only one nucleotide that separates these 2 genes. M and F, 1, F and HN, 35, HN and L, there are 47 nucleotides that separate the HN and the L. So over here, we have 1,746. This is the total genes for NP, total nucleotide for P, total nucleotide for M, HN and L. Alright, these are what it means by the total. Alright, so let's look at how the polymerase, like I mentioned to you, the polymerase is the one that responsible for the synthesis of this mRNA. Remember, the polymerase that synthesizes the mRNA for NDV come from the virus itself. Alright, it comes from the virus itself because the host doesn't have RNA-dependent RNA polymerase. Okay, please remember that. Right, so now let's start looking at how this mRNA is being synthesized. Right, I mentioned to you there are 2 nucleotides that separate NP and P. So how does the polymerase know this is the end and this is the start of the new gene and end of the previous gene and start of the new gene. Let's look at the sequence of NEV. So the mRNA, the characteristic of the mRNA, remember we mentioned that there's a cap and there's a poly A tail. And we also know that when you look at the whole mRNA, you will know that not the whole mRNA are responsible for, no, let me rephrase the sentence. Not the whole mRNA is the gene. So this is the mRNA for a gene, but it will only be translated into a protein when they see a start codon, where this will be the methionine. Then they will stop at stop codon. That's why we have at 5' end, 5' untranslated regions, and 3' end, we have 3' untranslated region. Alright? So here what we can see, this is a standard mRNA, 5' untranslated region with a cap, start codon, stop codon, and this is the region that is the untranslated region where it doesn't being translated into a protein, and we have our poly A tail at the end of the 3' untranslated regions. Alright, so now I'm using MP gene as an example. Okay, MP gene as an example. So the MP gene here, the coding region is 1470 nucleotide. So here, the first nucleotide that belongs to NP is 1746, whereas the gene that coded for NP protein is actually 1470. So the rest of the nucleotide fall for the untranslated regions here and untranslated regions here. Alright, so this is the mRNA for NEV. Right, now I mentioned just now, how does the polymerase recognize this is the beginning of a new mRNA and the end of the new old mRNA and where it should start? So upon analysis of the genome, what we found that the mRNA for all the gene will always start with UGCCCAUCUU. Alright, so this is the mRNA for NP, the mRNA for P, M, F, HN, and L. You will notice that all of them has identical sequence. So this is the sequence that let the polymerase know that this is the beginning of a sequence. What about the end? So now we have the start, then it will continue with untranslated regions, then start codon, stop codon, then untranslated regions, and towards the end, how does it know this is now the end and you should synthesize poly A tail? We notice that there is AAUC 6U at the end. 6U, 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 6U. Alright? So what happened here is the polymerase will recognize this sequence and start synthesizing a poly A tail. So what it means here is when the polymerase, let's go back to here, when the polymerase binds to the 3' end where it behaves like a promoter and start going into the NP, it will start recognizing this sequence and it knows that this is the beginning of a new sequence. So it will start synthesizing. When it comes to the end, when they see AAUC UUUUUU, you notice that there are 6 U here. What happened here is the polymerase will undergo a process called stuttering. STUTTERING. Stutter means that it's just like, you know, the playback and playback, you know, the CD that hang. So the the polymerase will go back and synthesize, go back and synthesize, go back and synthesize, continue this process for 20 times. So ultimately, what is going to be produced is it will produce a total of 120 A's towards the end of the mRNA. So we look at here, we have a cap, we have the NP, okay, from beginning to the end, and then it will end with 120 A's for the mRNA. So notice that 3' end, 5' end, this 3' end is being used as a template for the polymerase to bind to and start synthesizing the first mRNA for the NP. Alright, so this is how it looks like. And then when it synthesizes about 20 times to produce 120 polyaethyl nucleotide of polyaethyl, alright, what happened is now it goes into intergenic region. Okay, intergenic regions. Remember I said between the NP and the P, there are two nucleotides that doesn't belong to NP or P. So this is the two nucleotide. So when the polymerase come to here, they know that this is nothing to do, not synthesizing anything. But when then they go to the P, they will start synthesizing this as the first nucleotide and continue with the P gene and polyaethyl. So let's look at this in a little bit more detail by looking at this. I know you won't be able to see the sequence here, but if you can, please display this sequence in your laptop so that you can see it. So we started with the 3' end leader sequence. This is the leader sequence. Alright, this is the leader sequence. And remember we said the start of an mRNA, we call the NP gene start. Right? The NP gene start has this sequence. This is 3' end to 5' end. The genome has this UG, CCC, AU, CUU. But when you translate it into 5' to 3', it becomes AC, GGG, U is the T, A, okay, UA, GAA. So this becomes the gene start of the mRNA. And then the yellow color here, these are the sequence for the 5' untranslated regions. Alright, so here, where this is where the cap is located here, EIF4E binds to EIF4G, and then it will start scanning for ATG. Is there any ATG here? There's none ATG until it sees the first ATG, and when it sees the first ATG, what happened now is EIF5 and 5B will now digest the GTP of the EIF2, and then what happened? Disintegrate of all the EIF, and then ribosome will bind to it. 60S ribosome will bind to it and start synthesizing the first methionine. Alright, so we have our first ATG here, and it synthesize all the protein here. Right, so now let's look at it in the synthesis of mRNA. Don't confuse the two, because students tend to confuse between the translations of the ribosome and this. But now we are looking at the polymerase that recognize this gene start and synthesize from beginning. Synthesize. Okay, polymerase doesn't recognize gene start or doesn't recognize start codon and stop codon. Their main role is just to synthesize the nucleotide. Alright, so polymerase starts synthesize the gene start, the yellow color, 5' untranslated region, and coding sequence, and then you can see the stop codon here. Then it will also continue to synthesize the 3' untranslated regions, which lie in purple color, until they see UUA, GAA here, A, and they will repeat this for 20 times to produce 120 A. Alright, so this is the genome sequence. Alright, this is the genome sequence. So they will continue this, and eventually what we are getting is a poly A tail of the mRNA. It starts with this and end with 120 poly A tail for the NP. Right. So I also mentioned that the intergenic between the NP and P is 2 nucleotides and is CA. So CA is 3' to 5'. When you convert to 3' to 5' to 3', it becomes GT. So these are the two intergenic sequence. Now look, this is the new gene, which is the P, ACGGG, TA, GAA, ACGGG, TA, GAA. So now your polymerase recognize this is the beginning of a new mRNA, and it will now start synthesizing this as an mRNA, an ATG, and so on and so forth. So this is how polymerase for NDV to recognize the gene start, coding sequence, and gene end, and poly A tail. So the polymerase for NDV is quite powerful, whereby it will synthesize the mRNA as well as capping them and synthesize a poly A tail for the mRNA. So ultimately, what we are having is mRNA for NP, mRNA for P, mRNA for M, F, HN, and L. So in total, we have 6 mRNA that are responsible for the productions of the structural protein for NDV. Right? So I urge you to go back to the previous slides and look at it over and over again, and so that you really understand the concept of how mRNA for this virus is being produced. So now that the mRNA is being produced, what next? We have the mRNA that will be translated by the ribosome of the host to produce the structural protein, but we also have 2 non-structural proteins that is being produced by the NDV. The 2 non-structural protein, which is the V and the W. So bear in mind, V and W are not considered as structural protein because you can see there's no V or W protein in the virus itself, but it only occurs when the virus is replicating. Okay, so what we see here is, here we have NP, P, M, F, HN, and L gene. We notice that during the transcriptions of the P, or the productions of the mRNA, transcriptions of the mRNA for P gene, what happened here is during the polymerase synthesis, the polymerase recognized a region of the P gene in here, AAA, AAG, alright? So they recognize this sequence, and what happened here is after this sequence, they added an additional G. Alright? So this is P gene, P mRNA. They will just synthesize based on what happened in the genome, what is present in the genome, and they start synthesizing this sequence. But for the polymerase, what it done is when they see AAA, AAG in P gene, what it does is it adds an additional G into the mRNA. So now you can see with an additional of 1 G, it causes the frame, reading frame for the ribosome to be shifted. Here we have A, K, K, alanine, lysine, lysine, guanine, proline, tryptophan, serine, and serine. But when they add an additional G, it causes the frame shift, and now glycine is still the same, proline is still the same, but tryptophan has changed to methionine. So what we have here is we have the P and the V that has the same N terminal, but the C terminal is totally different. So W become M, serine now become valine and glutamic acid. So you can see the C terminal, the N terminal for these two proteins is the same, but the C terminal for these two proteins is totally different. This is because of an additional 1 G. So in normal conditions where the virus infects a cell, 65 to 70% of the gene for mRNA is P, right? But about 20% of the genes are the V mRNA, all right? So we have 60 to 70% of P, 20% or 25 to 30% is the V, and we also have another mRNA, which is the W. V additional of 1, W additional of 2G, right? Additional of 2 guanine. Here you have another G here that causes another frame shift, and ultimately what we produce is another protein that has the same N terminal as the P, but different C terminal from the P. So P in total has 395 amino acid, but V has 239 and W have 179. So this is expected because when you have reading frame that shift, it's very likely that the ribosome will encounter the stop codon earlier than the P, all right, or later than the P, but in this case, V and W has shorter amino acid, right? So they encounter stop codon way before the stop codon for the P. That's why P have 395, V have 239, and W have 179. Why is it so important for the virus to produce these two proteins? Now, like I say, the two proteins are non-structural. So it will only be important when the virus undergo replication. Sorry. So what happened here is V protein, now we have identified V protein is actually a protein that antagonizes the immune system in the host. For example, when the virus infects the chicken cell, the virus undergo replication, produce mRNA, and then they will produce the V protein. Now this V protein in normal cell, the immune system will react by producing interferon. This V protein is the one that antagonizes the interferon system in your cell that prevent the virus clearance from the host cell. So this is a very important protein that is responsible for the virulence of the virus. Whereas right until now, we have no idea what is the functions of the W protein. Right? We know they're present, but we do not know what is the functions of the W protein yet. Right? Hopefully one day we'll know. So in total, we have like 6 structural proteins plus 2 non-structural proteins that are being produced during the virus replications. Alright, so now translations happens, they produce mRNA, then they undergo the same process of translations in the host. And what happened? The MP, P, and L, these are all the proteins that will be produced and in the cytoplasm and will remain in the cytoplasm. M protein will be produced, but this M protein will interact with the cell membrane for assembly. So during the translations, the M protein will be produced, and this M protein immediately go underneath the membrane, and surrounding, and get ready for budding. Whereas this will continue to undergo transcriptions. Alright, we also noticed that the M protein, not only they will interact with the cell membrane, we found that some of the M protein translocate itself into the nucleus. Why? This is to modify ribosome and host shut off the host synthesis. See, the virus NDV also take over the transcriptions process or translation machinery in the host cell by translocate into the nucleus. What about the 2-glycoprotein? The 2-glycoprotein will be translated and then being processed in the rough ER. Okay, this is where in the rough ER, this is where the carbohydrate is being added. Remember glycoprotein? Yeah, they have glyco or carbohydrate that being added on the protein. Then from rough ER, they go to Golgi apparatus where the carbohydrate chains are being processed, added with skeleton. The same go for F, and then go through the Golgi apparatus, trans-Golgi network, all the way to the cell membrane, and now you have HN that being displayed on the surface of the virus, surface of the cell, and also the F on the surface of the cell. So here, all the protein are being produced, being processed. They are getting ready for the assembly. But we also need to produce the viral genome. So what happened here? 3' to 5' end. This is the viral genome. We know that the RNA is being used to synthesize mRNA. Okay, the viral genome is being used as a template to synthesize mRNA. So when does the virus know that they already have sufficient of viral protein and now they should undergo genome replications? So just to refresh your memory, for poliovirus, we have 3 CD that circularize the mRNA and prevent the translations to happen. But in dengue virus, we have an S5 that bind to stem loop A that stop the translations from happening. For NEV, the proteins that are responsible for stopping the translations and initiate the replication is the P protein. So you can see how important P protein is. Not only it protects the RNA, but it also the protein that stop the mRNA, stop the transcriptions of the mRNA, but initiate the replications of the genome. So it is believed that NP bind to the transcription complex. So if you remember previously, we have the RNA surrounded by NP, and this NP is associated with P and L protein to carry out the transcription process. So now when we have abundance of NP in the cytoplasm, this NP will now bind to this complex and initiate a replication. Tell the transcription complex that stop synthesizing mRNA. It is time for us to synthesize the genome of the virus. So what happened here? Polymerase will now start synthesizing the RNA. Remember, they recognized the sequence, the poly A here, and they started. But upon binding by the NP protein on the polymerase, what happened here is they will not stutter and continue to synthesize all the way until the 5' end. So in the end, what happened here is they are producing a genome complement, which is a 5' to 3' end. Alright, so now this is the complete positive strain RNA for NEV, which will now be used as a template to synthesize more of the negative strain RNA genome for the new viral progeny. Okay, now in the cytoplasm, we have a lot of mRNA that produce a lot of viral protein. In the process, we also have a lot of negative strain RNA that being produced during the genome replications. So what next? They can either, this negative strain RNA genome can either be used to undergo a secondary transcriptions. So this negative strain will carry out another round of mRNA productions. Now you have more negative strain. So now you have more negative strain, then you will produce more mRNA. This is what it means by secondary transcriptions. So you produce more mRNA and you will produce even more viral protein within the cell. So let's say now that all the viral protein has been produced abundant inside the cell, and then we have a lot of viral genomes. What next? The 5th step of the virus infection, which is the assembly. So the viral genome is always surrounded by nucleocapsid. Nucleocapsid surrounded the RNA. Then you have P and L, the polymerase, and phosphoprotein, the activate. So what happened here, this is the complete transcription complex that is ready to bud out. What next is, you can see under this is a cell's membrane. You have H and F that is already assembled, getting ready to bud out. You also have matrix protein that underlie the membrane. Alright, so once the MP interact with the interact with the M protein, this will start to initiate the budding process of the virus. Right? This interaction is very important. Otherwise, during this step, the virus can bud out. But it will not bud out until the M protein is interacting with the P protein, NP protein. So when M interact with MP, this is the time where it will initiate a budding process, as what we can see here. This is the membrane. They will start to bud out. Okay, starting of budding. They started to bud out, and eventually they will produce a virus that have the HN and the F on the surface and the transcription complex in the core of the nucleus. Now, during this budding, remember we have HN, which is hemagglutinin neuraminidase. The neuraminidase is very important during the budding, whereby the neuraminidase is the one that now cleaves the sialic acid receptor on the cell's surface, because you do not want the virus during budding, the HN bind to the receptor again. So here the neuraminidase will start cleaving all the sialic acid receptor on the surface, which allow the virus to be released from the host cell. Alright, so now we have finished the life cycle of Newcastle disease virus, where we start with attachment, fusion, how the mRNA is being synthesized to produce viral protein, genome replications, assembly, and now egress or budding for this virus. Now you will notice that of all the viruses that we have discussed so far are basically human diseases. Why am I discussing about Newcastle disease virus? So I would like to introduce you to conditions where we have a misconception that all the viruses are bad. But we also know that sometimes this virus can also be used as a beneficial virus. One example is we call oncolytic virus. Newcastle disease virus, or NDV, is also one of the oncolytic virus. What it means is that the virus can specifically target cancer cell, and it starts replicating. So example here, this is the cancer cell that present in your body, in the patients. So when the virus go into the host cell, they will start replicating and kill the cancer cell and being released. So what it means here is that the virus that is actually pathogenic to the chicken could actually being used as a virus that is beneficial to human. So you might wonder, well, you inject virus into my body, so will I not get sick? Remember, the virus is host specific. Of course, you also have genetic virus, but NDV is so specific is that it's only infect chicken and cause disease in chicken. If humans have the virus being injected in their body, for example NDV, they only have flu-like symptom, a very mild symptom, and you will recover because your immune system will get rid of the virus. But because in cancer cell, their cancer cellular immune system is down, that's why it allowed the virus NDV to infect them and start killing them. So ultimately what happened here is the cancer cell will eventually be killed by NDV. Then you ask, what about the normal cell? When the normal cell being infected by Newcastle disease virus because our body also have cellular immune system, and the V protein is only specific to the chicken cell. So in our body, even though they express the V protein, it will be killed by our immune system. So that's why your normal cell is unharmed. So this is a very interesting phenomenon that being used, and recently in 2015, the FDA has approved the first oncolytic virus to be used as the agent to kill cancer. So these cancers being treated by virus is skin cancer, melanoma cancers, and the virus that used here is herpes simplex virus. All right, so it's very interesting. They get a lot of good result from using oncolytic virus to treat cancer, because we know a lot of cancer has been resistant to the chemotherapy drugs, and you know those patients, some of patients who undergo chemo or radiotherapy end up having recurrent of the virus, recurrent of the cancers. So this is a very good alternative to be used for the treatment of cancer. So as a summary for this, we have looked at the negative-strand RNA, enveloped negative-strand RNA virus, and we look at the life cycle of Newcastle disease virus. We look at how it's being attached. There's two models, the H and F interacting or non-interacting at the beginning. Then we look at the fusions, the fusion peptide that binds, penetrate the host membrane, and then fuse the two membranes together. Then translations of viral protein, which in the beginning they need to transcribe the mRNA for the translation to occur. Then once the viral protein is abandoned in the cell, then MP protein will be binding to the transcriptase or polymerase and transcriptase that stop the transcriptions of mRNA and initiate the replication of the viral genome. So once it's being done, a lot of viral protein, a lot of viral genome, the next step will be encapsulation or assembly. So they will now assembly into a viral, almost complete viral genome, virus. Then you will now bud out from the host cell. Please note that the NDV, the enveloped virus, do not lyse the cell. They normally bud out from the cell. So that's it for this second part of enveloped negative-strand RNA virus. Thank you.